Welcome to our lecture on topic 2.4 carbohydrates. Our essential idea for today: carbohydrates are oxygen-rich biomolecules which play a central role in metabolic reactions of energy transfer. Our objectives: we are going to define and classify carbohydrates. We're going to look at ketoses and aldoses. We're going to look at linear and cyclic forms of monosaccharides, especially glucose. We'll talk about alpha and beta forms of monosaccharides, and then we'll look at some experimental methods to figure out if we even have a sugar in a sample. We'll describe examples of disaccharides, review condensation and hydrolysis reactions, which is how we get those disaccharides and polysaccharides. We will look at 1,4-glycosidic linkages and 1,6-glycosidic linkages. We will talk about some polysaccharides, and we will wrap up with a look at how to figure out if you have starch or not. So first up, what even are carbohydrates? Let's define them. So carbohydrates, literally hydrates of carbon, are going to have most of the time, not all the time, but most of the time, will have a 2-to-1 ratio for hydrogen and oxygen, and then we'll have fairly similar numbers of carbon than those waters, which is why we call them hydrates of carbon, carbohydrates. We're going to classify our carbohydrates into a couple different forms: monosaccharides, disaccharides, polysaccharides. Monos are the monomers, of course, dimers, and then polymers we'll talk about later. So our monomers, our monosaccharides, we can classify into a couple different categories. We have aldoses and ketoses. So our aldoses are literally aldehydes that are sugars. So notice that we have this carbon double oxygen bond here at the end of the molecule. That makes this an aldehyde since our carbonyl group is at the end of the molecule. So we call these sugars aldoses. Ketoses are ketone sugars. Notice that the double bond between carbon and oxygen is in the middle of the molecule, not at the end. That makes this a ketone because the carbonyl is in the middle. So we have aldoses and ketoses. We can also classify our carbohydrates based on the number of carbons that they have. So a triose would have 1, 2, 3 carbons. This is a triose that is also an aldose. We can have pentoses that have 5 carbons. This is ribose. It is also an aldose. And then glucose is a hexose. It's got 6 carbons in its molecule. Our monosaccharides can come in either straight chain forms or cyclic forms. Cyclic is way more stable, so we're going to see this more often than we'll see the straight chain forms. But it's interesting to look at how we get from straight chain forms to these cyclic forms over here. So this carbon right here, I'm going to focus on this carbon for a second. With this H and O, what that means is that we have a single bonded H and a double bonded oxygen. So this is one of our aldose sugars, an aldehyde sugar. And I'm going to also focus on this carbon down here. So not the very last one, but the second to last one. This carbon, I'm actually going to make it a special color. This carbon is this carbon right here in this picture, also this one over here. And then this carbon is here. Notice the double bonded O. It's here, the double bonded O. And so that oxygen, that double bonded oxygen might be oriented up or down depending on all kinds of cool stuff that's happening. But what's going to happen is we're going to end up with some bonding happening in this region, regardless of whether the double bonded oxygen is going up or down, up or down. We're going to end up with this lovely functional group that we're going to call an ether. It's an ether because I have carbon single bonded to an oxygen, single bonded to a carbon, no double bonds in there. It makes it an ether, not an ester. And then again, depending on whether my double bonded oxygen was down or up, I'm going to end up with hydroxyl groups that go down or hydroxyl groups that go up. And we'll talk in a couple more slides about whether this is alpha or beta. Actually, no, let's talk about it right now. So this is alpha glucose, and we know it because this guy right here, so look at like the little flag thing that sticks up on the molecule. So if this is up and this is down, if the hydroxyl group is away, then it is an alpha molecule. If they are by each other, hydroxyl group up and carbon up, if they are by each other, then it is the beta form of the molecule. So away for alpha, by is beta, and again, away, I'm looking at this guy up here and that hydroxyl group on carbon number 1 versus that flaggy thing here and the hydroxyl group, again, on carbon number 1. They are by each other, it's beta. They are away from each other, it is alpha. Good. These pictures are all known as Haworth projections. Our Haworth projections simply show no hydrogens, and our carbons are only shown by those vertices in the drawing. It just helps to reduce the clutter, so it's a little bit easier to see the shapes, the important interactions from special functional groups, as opposed to every single hydrogen that is all over this thing. So again, our aldoses, like glucose, aldose because that double bonded oxygen is at the end of the molecule. It's an aldehyde sugar, an aldose. So this guy is going to form this 6-sided ring. So I've got carbons, 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 5 carbons in the ring plus 1 carbon up here. We have that ether oxygen in here. Carbon number 1, 2, 3, 4, 5, 6. What is super cool about our ketoses, so our sugars that are ketones, because the double bond is here, this carbon is going to wrap around and bind to that double bonded oxygen. But because it's in the middle, not at the end, we actually end up with these 2 flags. So I end up with 2 little flags instead of just 1. Good. And because the hydroxyl group on carbon number 1 is by this guy right here, carbon number 6, then this is actually beta fructose. So here is the pieces of carbohydrate that we end up with in our data booklets. So our data booklets, table 33, right under our lipids, we have these carbohydrates. Notice that you have straight chain and cyclic versions of glucose. This is alpha glucose because here's our flag thing, here's our hydroxyl group, and they are away from each other. We also have straight chain fructose and alpha glucose. Notice that here I have up and I have down, they're away from each other. So this is alpha. We've got also the cyclic form of ribose and deoxyribose, which are the sugars that are in our DNA and RNA. So let's focus on glucose for a little bit. So glucose is the main product of photosynthesis, and it is the primary reactant in cellular respiration, and so it is so very important. It's the main component of many of our polysaccharides. We use it in fermentation reactions, so glucose is an incredibly important molecule, an incredibly important carbohydrate. Again, it's given to us in our data booklet, table 33. We get both straight chain and cyclic forms of glucose. Let's look at this super cool fermentation reaction that is shown here. So yeast, when they undergo anaerobic respiration, they are going to take glucose molecules and break them down into ethanol molecules, and then carbon dioxide is a product as well. So yeast can make ethanol. Ethanol, of course, is the alcohol that is in alcoholic beverages that you can drink when you are 21, but never in excess, my friends, because it's not good for you. It's also a biofuel that can be added to our gasoline to add a little bit more volume to our fuels. So these aldoses are also known as reducing sugars. Remember that aldoses are our sugars that are aldehydes, so they've got that carbonyl group at the end of the molecule as opposed to in the middle of the molecule, which would make them ketones instead. So our aldoses are known as reducing sugars because they are very easily oxidized. They are oxidized, they are reducing agents. Because they're sugars, we call them reducing sugars, but they're basically sugars that are reducing agents, right? So notice that the carbonyl group here can be oxidized using some nitric acid and some water to form a carboxylic acid. So we're going from aldehyde to carboxylic acid. This should sound vaguely familiar to you because a long time ago we definitely talked about the oxidation of primary alcohols. And if we distill out the product, we have an aldehyde, like that one right there. But then if we put it under reflux, we can complete that oxidation and get a carboxylic acid. So this is simply the very end of that process, kind of similar to that last step. So going from the aldehyde to the carboxylic acid, this oxidation sugar is acting as a reducing agent. We can also oxidize this other end, this flaggy part, that terminal carbon, and put another carboxylic acid over here. Pretty cool, right? And we actually have a couple different laboratory tests that we can carry out to figure out if we have reducing sugars, aldoses, like glucose in a sample. So 3 common tests that we have for aldehydes or aldoses, reducing sugars. So Benedict's test, we're going to reduce copper from a +2 to a +1. That copper will turn from this blue to this orangey color when there is a reducing sugar in the solution. So the sugar will be oxidized, the copper will be reduced. Failings is super similar. We're going to take this copper and it's going to be reduced down to a copper +1 because the sugar will be oxidized. And then one last one, Tollens, is super similar except it's silver instead of copper. And then we end up with this super pretty silvery mirror stuff going on inside of our test tube. Again, because the sugar has been oxidized and this metal, the silver, has gone from +1 reduced down to a 0. Let's put away our monosaccharides and talk about some disaccharides for a little bit. So disaccharides are our dimers of carbohydrates. They are composed of 2 monosaccharides bound together. 3 different disaccharides that we're going to focus on just because they're super common and digestible in our food. So sucrose, which is composed of glucose and fructose. So we have 1 glucose monomer and then a fructose monomer next to it. Lactose is galactose and then glucose, and then maltose is 2 glucose monomers bound to each other. Lactose, of course, is milk sugar. Sucrose is the most common sugar in our food. When you go to the grocery store and you buy sugar, it is sucrose. And then maltose is pretty cool. When we break down starch in our small intestines with amylase, we'll talk a little bit more about amylose in starch. So we have this enzyme amylase that will break down our starch. The first product is maltose, then we have another enzyme that breaks that maltose down into glucose, and that glucose is absorbed into our bloodstream and we use it for cellular respiration. So looking at these disaccharides, we're going to form our disaccharides from the monosaccharides via condensation anabolic reactions. And it's pretty similar to what we were looking at with our proteins, with our lipids. So basically what happens is we take the hydroxyl group of one guy, the hydroxyl group of another guy. We take one of those hydrogens and both the oxygen and hydrogen to make water. And then the oxygen that's left becomes this bond in the middle. So we have an ether. Again, it is an ether. It's a special kind of ether that we're going to call a glycosidic linkage because it's glucose and other sugars. So glycosidic linkage, kind of like we talked about those amide linkages in our proteins, glycosidic linkages in our disaccharides. We're going to call this particular kind of linkage 1,4 glycosidic linkage because this carbon is number 1, this carbon is number 2, 3, 4, 5, carbon number 6. This guy, again, oxygen, is here in the ether. I have 1, 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6. And so this is going to be a 1,4 glycosidic linkage because it's carbon number 1 of one monomer and carbon number 4 of the other. So 1,4 glycosidic linkage. Let's go ahead and chop this guy apart. So review hydrolysis reactions. We're going to cut that bond that we made on the previous slide. We're going to add some water to this guy. This hydrogen is going to jump onto that oxygen, and so now we will have an OH group here. The hydroxyl group here is going to jump right in here. We'll have a hydroxyl group here. This bond has been broken and disappeared. We now have 2 monosaccharides instead of a single disaccharide. Hydrolysis, splitting things using water, hydrolysis, and this of course would be a catabolic reaction as opposed to anabolic. This, just a little vocab reminder, because I didn't show the carbons in the molecule, I'm not showing all those extra hydrogens in the molecule. This would also be considered a Haworth projection. Good. So we're going to take a little detour here into some incredible science history stories and talk about sugar substitutes and their discoveries, their inventions. So this guy in 1878, this is Konstantin Fallberg, he was working in a chemistry lab at Johns Hopkins University, left work, went home, he was eating dinner, noticed that his dinner roll was extra sweet, realized that because he never washed his hands after he left his work laboratory, that whatever it was that he'd been working on at work was definitely still on his fingers and was transferred to his bread. And then his bread was super sweet and he was like, amazing. And so this is saccharin. This is how we discovered saccharin, is because this guy, Konstantin Fahlberg, didn't wash his hands after he left work. So anyhow, fast forward 1965. Surely 100 years later, in this century, surely chemists are washing their hands, right? Wrong. Meet James Schlatter. He was working in a medical research lab trying to develop drugs that would help ulcers. Got some stuff on his hands, licked his fingers in his chemistry lab, noticed it was super sweet, and voila, aspartame was discovered. Aspartame, of course, the fake sugar that is in NutraSweet and Equal, and thanks to James Schlatter licking his fingers in chemistry lab, we have this amazing substance. But my friends, please don't get any ideas about licking your fingers in our lab. We wash our hands. We do not lick our fingers in chem lab, ever. We're going to go ahead and focus now on some polysaccharides. So polysaccharides, because we are done with monosaccharides and disaccharides. Let's talk about some polysaccharides. So polysaccharides are going to have these monomer units that are repeated to an indefinite number of times. So that's this N, as we have some unknown large number of repeats of this monomer. And it can be something, this N can be something that's a couple hundred of these glucose monomers long. It could also be like 20,000 glucose monomers that are all stuck together into a single polymer. So these guys can get crazy, crazy big. We're going to look at a couple different ones. We've got cellulose, we've got starch, and we have glycogen. So cellulose in plants makes up their cell walls. It is not an energy storage polysaccharide. It is instead a structural polysaccharide and makes up the cell walls of plant cells. It's formed from beta glucose, so the hydroxyl group on carbon number 1 is by that extra flaggy carbon guy, right? So beta glucose, which us mammals really don't make very good enzymes to break down beta glucose. We much prefer to break down alpha glucose with our enzymes. So cellulose for us in our diets is really just fiber because we cannot digest it because of that beta glucose, that chain of beta glucoses. The bonds are 1,4, so we have just carbon number 1 to carbon number 4 of the next glucose monomer bound together. There are no branches, and we'll talk about this branching thing on the next couple slides. Cellulose is going to look like this, definitely a straight chain, don't see any branches. What's kind of cool though is that each of these lines of beta glucose monomers that are becoming this cellulose polymer, each of these chains will hydrogen bond to other chains and make these stacks, which are super strong, which is why it's such a good material for plant cell walls. Starch is actually composed of two different polysaccharides, amylose and amylopectin. Both of them formed by plants, both of them composed of alpha glucose. The difference is that amylose is a straight chain, and I know you might be thinking that's not straight, but by straight we mean it doesn't have any branches. So it does curl up into that helix shape, but it doesn't have any branches, unlike this guy that does branch, and it branches again, and it branches again. So amylose, no branching, is going to be straight chain that turns into a curly Q, only 1,4 glycosidic bonds, as opposed to amylopectin, and again these two guys mixed together to make starch. Amylopectin has 1,4 and 1,6 glycosidic bonds. Again, we'll look at these super closely on the next slide, which means that it does have some branching. Notice branching here. And so this is amylopectin, amylose, amylopectin. The combination of these two guys is starch and is the energy storage molecule for plants. Glycogen is super similar to starch, but is made by animals instead of by plants. It's basically the animal version of starch. Again, alpha glucose, we have 1,4 and 1,6 glycosidic bonding, and so there is some branching. It's actually far more branched than is amylopectin. So amylopectin just about every 20 glucoses we have a branch, whereas in glycogen it's like every 10 glucoses we end up with another branch. And you can see that in the picture, so many branches. Let's talk about those branches. So again, amylose has only 1,4 glycosidic bonding or 1,4 glycosidic linkages. Here is carbon number 1, carbon number 2, 3, 4, 5, carbon number 6. On this guy, I have carbon number 1, carbon number 2, 3, 4, 5, 6. Carbon number 1 here, carbon number 4 here is where we find our bond. And so this is why it is 1,4 glycosidic linkages. That's a little bit different from our 1,6 glycosidic linkages, which is where our amylopectin and also our glycogen can form some branches. So again, I have carbon number 1, carbon number 2, 3, 4, 5, 6. This guy, carbon number 1, carbon number 4. Carbon number 1, carbon number 4. So these are our 1,4 glycosidic linkages. We also have 1,6. Carbon number 6, carbon number 1. So the little flag guy that sticks up on the glucose with carbon number 6 is going to form an ether with carbon number 1 of some other guy. And because it's going off on the side, that little flaggy thing, it makes our branch points. So carbon 1,4 glycosidic linkages is going to be just a normal old super cool straight chain. Carbon number 1,6, we're going to end up with these super cool branchy things that pop off. So how do I know if I have starch? Starch, again, is the combination of amylose and amylopectin. How do I know if I have starch in a sample? So our test for starch is really just testing for amylose. What happens is we take some iodine, so iodine and more iodine. Actually, this guy will kind of break apart and we'll end up with I3, so triiodide ions, and these triiodide ions will kind of settle themselves right into the middle of the curlicue of amylose. So they just kind of slip inside like that. And when they do that, they turn from a brownish yellowish iodine color to this super cool blue purple color from this complex between the amylose helix and that triiodide ion that slips inside. So this is how we know if we have starch in a sample. We can also test for iodine. So if I want to know if I have iodine in a sample, then I can add some starch to it. And if the starch turns purple, then we know that we've got iodine in a sample. Good. And my friends, we did it. We classified carbohydrates. Most of the time they are hydrates of carbon, where we have carbon and then some water stuck to it. Not exactly water because it's like all spread out in hydrogens and hydroxides. Anyhow, we also talked about ketoses and aldoses. Ketos are ketones, and we're going to have those carbons, and there will be a double bonded oxygen in the middle as opposed to our aldoses, which are aldehydes, and have that double bonded oxygen at the end. We looked at those cyclic and linear forms of monosaccharides where a hydroxyl group kind of rolls around and finds the carbonyl group and they make a ring shape. Those ring shapes, the cyclic forms, are far more stable than are the linear forms. We talk about alpha and beta. So if I were to draw super quick some glucose here. So if I have a hydroxyl group that sticks up, the hydroxyl group is by that flaggy chunk of carbon number 6, then it is beta. However, if the hydroxyl group is away on the opposite side of the molecule, then it is alpha. We talked about ways to figure out if we have reducing sugars. Reducing sugars are those that are easily oxidized. They themselves are reducing agents. We have phthalings, we have benedics. They turn from blue to that reddish color. And then we also have the toluenes where we have the silver go from colorless to that super pretty silvery mirror stuff. We described examples of disaccharides. Disaccharides are maltose, which is glucose, glucose, and sucrose, which is glucose, fructose, and lactose, which is glucose galactose. We talked about condensation and hydrolysis. We've been talking about this for a couple lectures now. Condensation, anabolic, we're making bigger things from smaller things. Hydrolysis is the opposite. It's catabolic. We're breaking things down into smaller pieces. We talked about 1,4 glycosidic linkages. 1,4 where we have carbon number 1 stuck to carbon number 4. That will make a straight chain, no branches in our polysaccharide. 1,6 does allow those branches to start to form in a polysaccharide. We talked about those different kinds of polysaccharides. We've got cellulose, it's beta glucose, 1,4 only. We have amylose, which is alpha glucose, and 1,4 glycosidic linkages. We have amylopectin, which is also alpha glucose, 1,4 and 1,6 glycosidic bonding, and so amylopectin is branched. The combination of amylose and amylopectin is starch. And then, of course, we also have glycogen, which is the animal version of starch, 1,4 and 1,6 glycosidic bonds, which means that it has branches. And then finally, my friends, we also talked about how we can find out if we've got starch in some sample. We add some iodine. If it turns a blue-purple color, starch is there. You did it. Good work.